Hello. Today I'm going to give a solo Saturday morning NeuroSim meeting update. We're going to talk about a number of different features, but first I wanted to give an update on projects and what's been happening in the lab in the last week. So on Monday we had our DevaWorm meeting and a lot of interesting things there. Jayadratha went through the larval synaptogenesis dataset, and we discussed sort of the details of that data set and some of the ways he could apply it to his project. We also talked about simulations of early life and some of the ways you can simulate sort of the formation of cells and the formation of what are called nucleotide-like models, minimal models of nucleotides and things like that. And then we talked about the possible shapes of life. So there were someone did some experiments in the Wolfram evolution platform, and we discussed those results. So that was a pretty good meeting, and that's on our DevaWorm YouTube channel. The second meeting of the week was Friday, and we had the open source meeting, and we had project updates for Google Summer of Codes, where Google Summer of Code scholars are continuing to produce code and make progress on their projects. And then I followed a little bit up on the open washing that we talked about last week, talking about Meta's Llama model. And then Jesse gave an update on some of his activities. He's got some internships running through JoePro and some other things like that. Hello.
Hi, how are you?
All right. What do we dive into some readings and things like that? Okay, so the first thing I'm going to talk about today is on this BI reading group. So this is the Biological Intelligence Complexity group. And this was something that Jesse posted in the Slack. I think it was in the Cybernetics channel, because what they're doing in the BI complexity group is they're trying to go over different keystone papers in the history of like cybernetics and AI and brain science. It's just a wide range of different areas. They talked about I don't know, remember what the last paper we talked about was. But this paper was by Warren Weaver. It was from 1948, and we'll get into that paper a little bit in a minute. But this is sort of a screenshot of the Zoom group. So I don't know why they were face palming here. We had a discussion about it in the Slack. But they were basically talking, they'll read the paper, they'll invite people to sort of interpret the paper, and then you'll have the rest of the audience kind of ask questions. So this was the Weaver paper from 1948, and I'm going to read from the chat on the right. So there was a quote, "To what extent a property of the observer versus the system?" That was kind of, if you think about second order cybernetics, where their focus isn't just on the system, but on the observer of the system, and how the observer plays a role in the system's supervision and regulation. So they're kind of tying in this Weaver paper to some of these topics in cybernetics. Kevin Mitchell gives a observation here. I think it's relative to the predictability of the effects of manipulations. That's kind of the pragmatic payoff. Kamran Razvan, so much reminds of the simple book titled Flatland, which is interesting because the book Flatland is this book that was written in the 19th century, and it was about these creatures that lived in a one-dimensional land, and they would not be able to see what was happening at 2 dimensions. And then there's this 2-dimensional land where the creatures weren't able to conceive of anything in 3 dimensions, and so on and so forth. And so it's a nice exercise in thinking about how, if you are living at a certain sort of dimensionality, you would be unable to perceive things happening at higher dimensionalities. And it seems kind of silly to think about it. But it's very important for things like visualizing data sets and phenomena of high dimensionality. So, like a lot of machine learning, one of the things that underpins a lot of machine learning today is taking a lot of complex data, taking up a very complex latent space, and boiling it down to maybe 3 dimensions that we can visualize. And so, in doing so, we usually throw out a lot of detail, a lot of information that is inherent in those higher dimensions. So it's kind of, I don't think people talk about Flatland enough, really. And so, and then this is another comment. We are always living in a flatland as we see what we see, not what is out there. Not understanding it means we are missing a dimension. So, again, like, there, some theories of cosmology posit 11 dimensions of space time, and we live in 3 of those, and, that's basically what we have to work with. And so that's what that's referring to. But it's also referring to the whole machine learning aspect of dimensionality reduction. And then there are, of course, these issues of sort of the curse of dimensionality and other things like that. Then someone says 100%. And then Richard Lang says, don't rock the gravy train either. But why would any observer agree that it is complex? Or any computationally constrained observer, perhaps? So that's Richard Lang. And then I think Jesse said the system will tell you if it's complex. And then Kevin Mitchell's responds, yes, I think the thing about complex systems is that they're not flat. So this is, again, this idea of flatland kind of came up in this commentary. And then I like the great facepalms theme. There's Picard as doing a facepalm. There's the great double facepalm of Picard and Riker. And then, of course, Darwin giving a facepalm. So it's a very common thing. This is the paper Weaver, Science and Complexity from 1948. This is where it gives this typology of different types of complexity: organized simplicity, organized complexity, and disorganized complexity. So organized simplicity is where you have this sort of, focuses on regularity. So in systems and behaviors and in relations, you have these like arrays, these ordered arrays of things. So it could be like the checkerboard pattern, or a simple square matrix, or some sort of locally connected thing that has only local connections. And then the dynamics of organized simplicity or sine wave, which is a periodic sine wave. And then going to organized complexity, you start to get these clumps or clusters of things. So in the systems, you get these clusters of things that are distinct, yet they're individual subunits. The behavior of organized complexity are sort of these, again, clustered behaviors. They begin to become aperiodic. They exhibit this sort of structure. And then you get the relations, which you see in a network here, instead of this locally connected lattice. You get this network that has modules in it, and you get this branching that leads to the modules. And then the dynamics are, you get shifts in the dynamics that represent these kind of clusters. And then disorganized complexity is where you have, disorganization. So one of the things about this paper, of course, is that it comes right or well before the era of chaos. And so there's this whole field of chaos theory, but also this idea of operating on the edge of chaos, and where you have ordered systems that become more disordered as they become more complex, but they still maintain some sort of general order. So they can sort of be poised on the edge of chaos. They can be poised to undergo phase transitions at any given time. They're more sort of excitable. They can change their state more readily, but they still have this organizational aspect to it where they're not completely random or completely out of control, I guess is the way to put it. And so this is the language is different here, but it's kind of a similar concept. So for disorganized complexity, you have systems. The systems sort of are atomized. The behaviors are sort of maybe on the edge of chaos. It looks like you have a cellular automata here, where you get a lot of things that kind of regenerate, patches that are varying sizes. And then the relations are this complex network. We're heading from in organized complexity, we have sort of this ordered network of modules, this branching, and then you go to this what is sort of approaching a hairball network, is what they call a hairball network, which is where you get random connectivity but interspersed with ordered connectivity. And then you have the dynamics, which are stochastic, but also have components that are, where the information lies. So in organized complexity, this trace has information in it. Disorganized complexity, this trace also has information in it, but it's embedded in a lot more noise. And so this is where you get to the edge of chaos. So one aspect of this discussion that the BI complexity group was talking about was that this comment by Cameron Razvan, we are always living in a flatland. We see what we see. And this, of course, goes back to the 19th century book Flatland, as I said. And so this is a teaser for some work that's coming up for our work from our group for the BICA conference this year. So the BICA conference is coming up next month, and we're going to be presenting a paper. This paper talks about super performers, which is something we did a paper on in 2023, and linking that to world models and intelligence augmentation. So this is an area that we've been exploring in our phasic research area. And so if you're interested, look up phasic on our website and you'll find out more. But in this paper, we actually talk about Flatland and how what we call a world model for an agent actually can be influenced by thinking about Flatland and then thinking about how to break out of Flatland. So there was this mention of Flatland in our paper for the conference here. And there's a citation which I'm going to talk about in a little bit. This is the Flatland fallacy, moving beyond low-dimensional thinking. This is a paper from Topics in Cognitive Science in 2018, and I'll talk about that paper in a minute. But I have a slide for the upcoming talk for BICA. So this lays out the Flatland problem for world models. This is a picture on the right of Flatland. It's stylized after Edwin Abbott's 1884 book, Flatland, A Romance in Many Dimensions. And in that book, he talks about these beings that live in one dimension or two dimensions, and they're constrained to that world. So beings that live in one dimension are on this basically like a number line. They can't move past one another. They can only move in their space. They have no access to two dimensions, so they can't eat anything. And they're just basically line segments moving along a larger line segment, constrained by other line segments. If you go to a two-dimensional world, you can have creatures of different shapes, but those shapes are two dimensions, and they can't access the third dimension, which is depth. So again, they can live on a two-dimensional plane, but that's about all they can do. If you think about early video games where they had this two-dimensional screen, it might have been a toroidal world where they just had access to the surface of the toroid and nothing more. So they could make movements along that plane, but there was no depth. And then 3D space, of course, is where we live. You have three-dimensional space. You have depth, but you also have width and height. And that's where we move. We move in 3 dimensions. Some people use time as a 4th dimension, but in terms of space, we have access to 3 dimensions. Now, we know from cosmology that they're higher dimensions, of course, but we can't access those directly. And so this is an image where you see this 2-dimensional character sort of rise above a plane or fall beneath a plane. And this 2-dimensional object is able to see the being only as it's crossing that plane and coming above it. So they can't see below. There's no depth. And so this is something that Carl Sagan talks about in his Cosmos series, where he has a video where he does this Flatland sort of demonstration of creatures being in 2 dimensions and creatures being in 3 dimensions. And then the implication is that you could have something in higher dimensions that we as humans couldn't perceive fully. So that's the idea here. And so for world models, though, there is this Flatland problem, which is that the world always has a higher dimensionality than the model. So if we have a world model for agents, that model has a certain dimensionality, which, might be an embedding, it might be a formal representation. And, we take things from the world, we kind of abstract them away from the world, and the world always has a higher dimensionality than the things that we abstract away. So unless we have every single aspect of the world in our latent space or on our representation, then, our world is always going to be more complex, or at least have a higher dimensionality than the model of the world. So again, this is where the paper comes in, Topics in Cognitive Science. And they talk about in that paper that there's this dilemma of Flatland, the Flatland problem in cognitive science. One part of this is that the thing that we use to really kind of deal with making scientific hypotheses out of complexity, something called parsimony, which is the simplest explanation, which is often the best explanation. But what we tend to do in making hypotheses is we tend to boil things down to the simplest explanation. But in doing so, we also lose dimensionality. And in fact, parsimonious explanations are inherently low-dimensional theories. They reflect reality of a much higher dimensional problem. So you can have a parsimonious explanation that deals with much higher dimensional spaces, but it may not fully describe those. Similarly, we can have simplified explanations of complex phenomena. We call that the Flatland fallacy, where we assume that the simplest explanation is the best, but sometimes we're just fooling ourselves as we inhabit this flatland. All right, the last thing I want to talk about here is this Topics in Cognitive Science paper. This is by Eshen Jolly and Luke J. Chang. And again, they're coming at this from a cognitive science point of view. So we have this abstract that reads, psychology is a complicated science. It has no general axioms or mathematical proofs, is rarely directly observable, and is the only discipline in which the subject matter is also the tool of investigation. Like the Flatlanders in Edwin Abbott's famous short story from 1884, we may be led to believe that the parsimony offered by our low-dimensional theories reflects the reality of a much higher dimensional problem. Here we contend that this Flatland fallacy leads us to seek out simplified explanations of complex phenomena, limiting our capacity as such to build and communicate useful models of human psychology. We suggest this fallacy can be overcome through A, the use of quantitative models, which forces us to formalize our theories, and B, improve quantitative training, which can build new norms for conducting psychological research. So their interest is in psychology very specifically, and they give some recommendations for overcoming this Flatland problem. And so, in they talked about that in the discussion group, that this is a problem that is sort of parallel to this idea of simplicity versus complexity. And in dealing with complexity, we tend to simplify things, but in simplifying things, we end up running into this flatland problem. So again, this is the Flatlander view of a sphere. It's a circle, it's 2D. The 2D creature cannot perceive the 3D. And in reality, we have this sphere in 3D. Actually, in reality, we might have a higher dimensional structure, but we can't perceive that as such. So this is just kind of a basic idea about this problem. Human psychology is rife with complexity. We're dealing with the brain, we're dealing with behavior. And, why does it happen? Why does the flatland fallacy happen? It's because it's a bias and a limitation of human cognition. So we have a lot of sort of four main reasons why this fallacy occurs. The first is that bias sees in the feeling of understanding. B is limitations of human cognition. C is over-reliance on traditional experimental design and analytical approaches. And D is limitations in our ability to communicate complex concepts. So you may be saying, well, how does modern complexity theory fit into this? And I think it's worth noting that this is something that's been talked about extensively in modern complexity theory, especially people interested in social science and complexity theory, where part of this is about experimental design flaws or limitations. Part of this is about our own cognitive limitations and biases of our home field or of making things easier. And so it's not as easy as you'd think to overcome this. It's not just a matter of applying the techniques of complexity theory. For one thing, those techniques are still lacking in terms of what they offer. But moreover, it's hard to actually deal with these problems, deal with these issues effectively. So this is the paper here. So Warren Weaver, as it turns out, he wrote, if you know the mathematical theory of communication, he's a co-author on that book with Claude Shannon. Claude Shannon was a researcher, a theoretician, and he worked at Bell Labs, and he did a lot of things. One of those things was develop the mathematical theory of information, or the idea of information entropy. And so you have this equation that's sort of famous. It's the H value for information. And so if we go to the board here, I'll draw out the equation so we can appreciate it. This is H equals, and it's usually minus summation, and it's a probability of I, and then there is this multiplier log probability of information. It's usually the way it works. Sometimes you can take this as log 2. Sometimes you can take this as log 10. Sometimes you can take this as natural log. And the difference here is that it's just the way in which you're normalizing the information. So if you have like a base 10, it usually gives you an index from 0 to 1. If you're doing it at base 2, it usually means you're dealing with bits. So sometimes people like to take different bits in a string and calculate the information, and that gives you that number. Log 10 will give you, or base 10 will give you an index between 0 and 1 on a base 10 scale. So if we're looking at any sort of integer, then that's what we get. And then log E is just, a sort of a natural log, a way to normalize it against a range of values. So the idea is that you can calculate the information. You'll usually have some ensemble of things. So the ensemble will be a number of units that you're looking at, and they have a certain amount of information. So for example, we have a bunch of circles here, and these circles are lights that turn on and off. And we measure that over time. So over time, and we want to know, how we don't necessarily want to know you know, if they're all on or all off. What we want to know in this ensemble is what is the pattern of on and off over time. So let's say we measure away. So we'll go for 100 time points. And we'll say that it's 40 on and 60 off. 60 off. And what we want to know is not like if it's 100% on or 100% off. What we want to know is what is the variation here. So if we have 50 on and 50 off, that's the maximum amount of variation over that time interval. So it's on and off, like basically at random. If we have 40 times on and 60 times off, that means that it's off more than it's on, and that means that there's less variety in terms of it being switched. So that's basically how this works. And then of course, if you go from, like, if your 100 is like 0 on and 100 off, that means there's no variety at all. That this is total, that we know that there's absolutely no information in this light or in this point because it's always on. So we can compress that information and ignore it. And what we want to know is basically in this array, how many of these go on, how many of these go off in their frequency. So if we go back to our equation, what we're going to do is we're going to plug in pi if we go back to our equation here, this frequency is going to plug into pi. So we plug 40 and 60 into pi, and I guess it could be like 0.4 or 0.6, depending on how we calculate it. I don't know. Maybe it's actually 0.8 because we have the divide 40 into 60, depending on how we calculate this probability. And then we take the log of that probability, so we normalize it, and then we multiply it by that same probability as a normalization procedure. And then for all of these lights, we know the same information, so that's where the summation comes in. Every light, we do the same thing, and then we get our H value. And as it turns out, we need this negative to normalize things. So that's where we get this. And then, of course, we get our information. So our information is here, H. And in Shannon's view, H is sort of, he called H entropy. All right. And so this is going to be problematic, perhaps, because we're not, if you think about like thermodynamic entropy, like if I have a cup of coffee on a table and it's hot, and then I leave it there for like 6 hours, it becomes cold, and it's said that the heat is lost through this process of entropy. So heat escapes and entropy happens, and but that's not necessarily what we're describing here. So why does he call it entropy? Well, there's a story behind that, which is that he basically chose that name because it was catchy and he could win debates using that name. It sounded impressive. But a better answer to that is that sort of the apocryphal story. The better answer is, first of all, you can, because of this ensemble nature, you can calculate things like H max, H min. You can actually get more information out of this this measure H, and it gives you this sort of property of calculating an ensemble of states, or evaluating an ensemble of states. Of course, the other answer is because there is actually an equation for thermodynamic entropy, and it is, if I can remember from memory here, wait a minute. Something like this. If I translate it into something analogous to H, it's S, and then you have this constant, and then you have the natural or transform, and then this probability. And what you're doing here is you're calculating this as an ensemble of microstates. So the idea is that in energetics, you have molecules that are all in these different microstates. You're finding the probability of any one set of states. So you have any ensemble of molecules. The molecules themselves can be in different states that evolve over time, and you want to calculate those out, and you get that. And then you have this energetic constant K, which is Boltzmann's constant, and then you have S. And so S and H actually are semi-related, right? I have a video on this on the YouTube channel where I go over some of these different equations. I go over thermodynamic entropy. I go over Shannon entropy, as some people call it, and kind of how they're related. Now, they're not related necessarily directly, but they have the same structure. They have the same sort of properties, and they're both dealing with these ensembles. So that was a long discussion on Claude Shannon and how this is he's relevant to this area. And then Warren Weaver was a co-author on that book, and I think he wrote the foreword. But Warren Weaver was a science communicator at the time, so he did a lot of stuff with kind of proto-complexity theory. So he was interested in cybernetics and sort of how that serves as proto-complexity theory in a lot of ways. So this paper is then by Warren Weaver, who talks about science and complexity. And so let's get to some meaty things in this paper. The first statement is how can we get a view of the function that science should have in the developing future of man? That's very 1940s. How can we appreciate what science really is, and equally important, what science is not? It is, of course, possible to discuss the nature of science in general philosophical terms. Let us, as a very realistic politician used to say, let us look at the record. Neglecting the older history of science, we shall go back only 3.5 centuries and take a broad view that tries to see the main features and omits minor details. Let's begin with the physical sciences rather than the biological, where the place of life sciences in the descriptive scheme will gradually become evident. So this is where he starts to talk about simplicity and reductionism and things like that. So in the 17th, 18th, and 19th centuries, the physical sciences learned variables, which brought us the telephone and the radio, the automobile, the airplane. The concurrent progress in biology and medicine were also impressive, but that was of a different character. The significant problems of living organisms are seldom those in which one can rigidly maintain constant all but two variables. Living things are more likely to present situations in which half a dozen or even several dozen quantities are all varying simultaneously and in subtly interconnected ways. Often they present situations in which the essentially important quantities are either non-quantitative or have at any rate eluded identification or measurement up to the moment. So what he's getting at here is that, he's making this sort of distinction between physics and biology. And physics, the idea is that you can do these controlled experiments where you can isolate variables. So you can have you can measure a single variable, and that single variable can tell you something about a system, and sometimes that is law-like. So it gives you this thing that you can just say, okay, this is an indicator of this other thing, and if I manipulate it in certain ways, it almost has this sort of causal linearity with the other thing. So I can create laws that apply universally. That's the idea. And so that allows us to develop technologies based on those manipulating single variables or multiple variables. And we know causally what will happen. It's very easy to understand that. In biology and medicine, however, what he's saying is that there are a lot of different variables that interact. There are a lot of different variables that have to be, they get modified in different ways in some process. So, like if you're looking at a physiological process or some process of life, you can't just manipulate one variable and get a strong effect on the other end. And then that's something that you can say is universal law. And so this is, again, just a broad stroke of the history of science. I don't think necessarily that, that it applies in all cases, but that's the sort of the comparison he's making. And so he gets into this issue of how biology up to about 1900 was about what people like to call stamp collecting, which is where you do collection of data, description, classification, and observation of concurrent or correlated effects. So, in physics, you can have like this, and of course, he's not talking about quantum physics here so much, or other types of nonlinear physics, but he is talking about sort of basic physics and how you can have the sort of the causality comes more easily in these kinds of systems as opposed to in biology and medicine. I'm not quite sure I agree with this. I think it's maybe that we didn't have the tools in biology and medicine, and we had sort of physics evolve with mathematics more closely. I don't know. I mean, like this is again something you would be able to discuss in the group here. So, to sum up, physical science before 1900 was largely concerned with two variable problems of simplicity, whereas life sciences, in which these problems of simplicity are often not so significant, have not yet become highly quantitative or analytical in character. Again, this has changed over the last 70 years or so since this paper was published. But this is the sort of the view that I think a lot of people have had up until very recently. So he gets into problems of disorganized complexity. So he brings up Josiah Willard Gibbs, who talked also about thermodynamic entropy. There's also a quantitative measure called Gibbs entropy as as a way to kind of deal with this is sort of where we're going from like these predictable quantities to sort of nonlinear physics, because we're starting to deal with dynamical systems, and we're starting to deal with sort of the foundations of dealing with the evolution of physical systems. So that's kind of where we're getting into disorganized complexity. So you remember that typology I showed you before I got into the paper? We have these three different categories, and this is where we're getting into those latter categories. So if we look at sort of what physics is done, in its history, rather than study problems which involve two variables or almost three or four, some imaginative minds went to the other extreme and said, let us develop analytical methods which can deal with 2 billion variables. So this sounds kind of familiar. This sounds like what we're doing in machine learning and deep learning, where we could do data analysis on 2 or 3 variables at once. But another way to go is to build like a generative model or a discriminative model that's of high dimensionality. And let's see if we can find patterns in that, or find some sort of causality in that. So we go from like the world of 1 and 2, maybe up to 4 variables, and we go up to different types of like very large or high dimensionality methods. That is to say, the physical scientists, with the mathematicians often in the vanguard, developed powerful techniques of probability theory and of statistical mechanics to deal with what may be called problems of disorganized complexity. So going back to our equation, this is sort of the foundation of statistical mechanics. This is Boltzmann entropy, And the idea of statistical mechanics is to deal with sort of these ensembles and look at their evolution over time. And the idea is that entropy is where you go from a very high amount or high degree of variation and variety in that thermodynamic ensemble to a very low level of variety. So things settle down. Energy excites my cup of coffee and makes it hot. When you lose energy, the molecules settle down into a state of coolness. So this is where, we get this sort of connection with the mathematics. And of course, that's just one small example of the mathematics. We get all sorts of different ways we can calculate large systems with a large number of variables. So that's, we're using statistical mechanics, we're using probability theory to deal with things called disorganized complexity. And of course, this was in 1948, so we would have to wait a couple more decades to get sort of the dynamical systems treatment of disorganized complexity, which led us to chaos theory and sort of this, some of these other categories of complexity that exist. So, this is again sort of setting the table for complexity theory. And so the classical dynamics of the 19th century was well suited for analyzing and predicting the notion or the motion of a single ivory ball as it moves about on a billiard table. So this is the billiards model of physics, where if I have, a cue ball, I can put it on the table and I can hit the cue ball and it hits another ball in play, and then, it might hit other balls, and those balls bounce off the edges of the pool table and might go into a pocket, and then you can actually perhaps predict the dynamics of that billiard table from the sort of the initial striking of the cue ball to the motion of that cue ball, how it strikes the other balls, and so forth. So you could predict the dynamics of a pool table. And it's, classical dynamics. There's nothing really stochastic about it. There might be, imperfections in the surface of the pool table. There might be imperfections in the balls. There might be, differences in how different people hit the cue ball. And that becomes important because, in the classical dynamics model, we don't care about those things. We just care about sort of the equations of motion and maybe entropy. And that's it. But then once we start adding in entropy, we start adding in these things that are sort of contribute to the net dynamics. By that I mean, like, you have this perfect model of sort of the equations of motion, but then there are things that impact that. And so you start adding in things like entropy, things like individual differences of the pool player, things like the surface of the table, things like air pressure. And some of those things play a very small role, but they're cumulative and they can add up. So he brings up this idea of the billiards analogy, where you can actually make perfect predictions of a billiards play by looking at the equations of motion. One can, but with a surprising increase in difficulty, analyze the motion of 2 or even 3 balls on a billiard table. There has been, in fact, considerable study of the mechanics of the standard game of billiards. But as soon as one tries to analyze the motion of 10 or 15 balls on the table at once, as in pool, the problem becomes unmanageable, not because there is any theoretical difficulty, but just because the actual labor of dealing in specific detail with so many variables turns out to be impractical. So this is interesting because this actually suggests that not only do you have this issue of sort of the net dynamics, but you also have this issue of computation. And that is like, again, this is in 1948, but you don't have the computational power to necessarily analyze every ball on the billiards table. You could only analyze 2 or 3, and you have to analyze, of course, the equations of motion for the entire game of billiards, so you can't analyze every ball, at least by the computational standards of 1948. Now, this is where we get into the modern sort of world of supercomputers, the supercomputers we have today, and even like predictive machine learning models, where we could feed a huge amount of data into the model, not just the equations of motion, and maybe predict what those trajectories would look like. So it's an interesting kind of point of computation here, where we have this sort of limit that might not hold anyone. So, but he goes on. Imagine, however, a large billiard table with millions of balls rolling over its surface, colliding with one another and the side rails. The great surprise is that the problem now becomes easier, for the methods of statistical mechanics are applicable. So now we can use different techniques. We don't have to just use the equations of motion. We can use statistical mechanics as a theoretical tool, and we can say, we can model this as an ensemble instead of just tracking each ball. To be sure, the detailed history of one special ball cannot be traced, but certain important questions can be answered with useful prediction. So some of those questions are, on the average, how many balls per second hit a given stretch of rail? Or, on the average, how far does a ball move before it's hit by some other ball? Or, on the average, how many impacts per second does a ball experience? So again, and notice that there are 3 words at the beginning of each of those questions, on the average. So what we do in both information entropy and in thermodynamic entropy is we're interested in this ensemble. We're interested in this group of things, and we're interested in either summing them or, calculating sort of the log transform of them, and then, using a physical constant to look at the overall state of these things. So we're always using sort of an average of the ensemble or a summary of the ensemble to get an answer. We're not interested in the individual behavior of these things, although that can be important as well, but we're interested in the overall summary. So like if you wanted to measure, say for example, the state of a pool table, the state of a pool table would be where you have a pool table and you have the pockets here. So you have like these pockets. And I'm just trying to kind of remember what a pool table looks like. And then you have your cue ball, of course, and you have your other balls scattered about the table. And what you're interested in isn't like a single strike of this ball and then there's a trajectory here, or might hit this ball and this ball goes here. You're interested in the state of all of these balls simultaneously, and then what is the mean behavior of these balls. So the mean behavior of the balls is eventually they all end up in a pocket, and you can actually get the mean behavior from the set of trajectories that happen every time you take your cue ball and you hit the other balls and they end up in the pockets eventually. What is the mean behavior over time of that? And then that actually can also give us information about that system, about the playing the game of pool. So it's a different type of prediction, different type of information. Okay, but this is interesting because you're dealing with either individual cases and sometimes individual variation versus averages of the variety. So you're doing maybe with individual, like distinct varieties of things versus this overall variety. Okay, earlier it was stated that the new statistical methods were applicable to the problems of disorganized complexity. So how does the word disorganized apply to the large billiard table with the many balls? So it applies because the methods of statistical mechanics are valid only when the balls are distributed in their positions and motions in a helter-skelter, that is to say, disorganized way. So again, if we go back to our pool table, the pool table often, we start off with our balls in sort of this, they're racked in this triangular shape, and then the cue ball hits that and distributes those balls all over the table. Eventually, you keep hitting them until they get into the pockets of the corners. But, so you go from this sort of ordered state where you have a pool table. And that pool table, you have the balls that are racked. Let's build this up here. So you rack the balls up here. They're all kind of in this. And then you have the cue ball. You hit those. Those break in different directions. Right? You break the group of balls, and then they go off into different directions, and there's like a lot of heterogeneity in how they kind of move and how they kind of distribute themselves and disperse. And then they end up in their homes, which is minimal entropy, because if they're in the pockets, we know exactly where they are, so we just need to find, how many balls are in each pocket, and we can describe the entire system. So pool is this really interesting system where we go from order, total order, to disorder or partial disorder, to order. And the order, of course, is not total order, so this is like initial order. And then we go to partial disorder, because it's not entirely disordered. I mean, they're not, spread out with maximum randomness. And then we go to the final order, or maybe the entropic order, which means that it's like they're in their destination. There's, there's variation, but there's limited variation, and it stays there. It's not going to pop back out of the pocket, So that's an interesting system. And so, again, this has parallels with complexity theory, with chaos theory, and so forth. So, let's see. For example, the statistical methods would not apply if someone were to arrange the balls in a row, parallel to one side of the rail of the table, and then start them all moving in precisely parallel paths, perpendicular to the row in which they stand. Then the balls would never collide with each other, nor with two of the rails, and one would not have a situation of disorganized complexity. So if you had a way to sort of move the balls in a very ordered fashion to their pockets, if you you had the balls in their rack, and then you broke the rack, and you had a way to line them all up on the edges and then put them in the pockets, that would be not disorganized complexity. That would be order just kind of being rearranged. From this illustration, it is clear what is meant by the problem of disorganized complexity. It is a problem in which the number of variables is very large, and one in which each of the many variables has a behavior which is individually erratic and perhaps totally unknown. However, in spite of this unknown behavior of all the individual variables, the system as a whole possesses certain orderly and analyzable average properties. So again, our trajectories are unique. They're individual. They're they have their own sort of way of getting to the pocket. But if we describe this pool table as an average, then we can see this pattern of moving to the pockets. It just it's at maybe a certain average rate for the ensemble. And then we could analyze a pool game like that. We could analyze a pool game in two ways. We could have a pool game that's analyzed by looking at the player hitting individual balls, getting into the pocket, looking at how easily they kind of do that, and then you think, well, there are all these variables that are intervening, and it's hard to really average across the balls because they're different distances to the pocket, and there's different things going on the table. There are differences in the player, and it just makes it unwieldy. The other way to predict it is by just taking the ensemble, taking its average, and then describing the average state of the game and saying, okay, if this player does this thing and they play on this table, the ensemble average varies by a certain amount, and then we can say, okay, well, we don't really care about the individual trajectories, we care about the outcome of the game, so we can become more efficient at our game of pool by maybe changing some strategies that we use, and overall it should help us be better, but not in every case. So he's kind of getting into this disorganized complexity idea and how you're dealing with complex systems as a manner of statistical analysis. You can deal with individual variables, you can deal with individual cases, and then average those across every case, and take a mean and a standard deviation and say some things about maybe statistical significance. Or you can use this ensemble approach where you're just analyzing the entire system on its average, on its mean level, this sort of mean field model. And then you're saying things about the average behavior. And then if individuals deviate from that average behavior, then at least we have a baseline for how they're supposed to behave. So, neither one is ideal, but I think the ensemble approach, what he's saying is that it's better for a problem like analyzing pool. Okay. A wide range of experience comes under the label of disorganized complexity. The method applies with increasing precision when the number of variables increases. It applies with entirely useful precision to the experience of a large telephone exchange in predicting the average frequency of calls, the probability of overlapping calls of the same number, etc. It makes possible the financial stability of a life insurance company, although the company can have no knowledge whatsoever concerning the approaching death of any one individual. It has dependable knowledge on the average frequency with which deaths occur. So when an actuarial table is built, they kind of take the age and they calculate out the the average likelihood or average probability of someone dying at that age. Now, someone, say, who's 80 years old, may be very energetic and healthy. They may be very unhealthy. But the average tells them what to sort of charge them. So in the aggregate, they can, make money off of those policies. So they're going to lose money on some 80-year-olds. They're going to gain money on other 80-year-olds. And it's just a matter of playing the odds on that. I know that doesn't sound very sympathetic, but that's the way that insurance and actuarial science works. So but they're, again, in that case, they're using this ensemble approach. Okay, so then there's this problems of organized complexity. So this problem of disorganized complexity, which is an advance over the earlier two-variable methods, leaves a great field untouched. One is tempted to oversimplify and say that scientific methodology went from one extreme to the other, from two variables to an astronomical number, and left untouched a great middle region. So again, with ensemble averages, we can go to very large numbers of things and calculate their average. But of course, that's not, the more we average, the more we kind of assume this sort of median behavior. And for the more, if we do this for more and more objects in our ensemble, we obscure a lot of variation, a lot of variety. And so that can be bad if there's heterogeneity in your system. What do? You can't just say the average works all the time, because it doesn't. There are a lot of things that deviate from the average. And if you have these clumps or clusters, like we saw with organized complexity, those clumps and clusters you're just averaging over those, and you have this mush of information. So you don't have the proper amount of information. So when we apply H prime and we apply, we do our sort of summation of our ensemble, we're kind of selecting our group. So sometimes our ensemble is very large and there's a lot of structure in that ensemble, but we're not capturing it with the average because we're, getting the information for something that's larger than that group, and we're washing it out. So I guess that's kind of the point there, is that, we can oversimplify things and end up with we're in the same boat as we would be if we were just using the two variable method. So the problems in the middle region, in fact, will often involve a considerable number of variables. The really important characteristic of the problems of this middle region, which science is as yet little explored or conquered, lies in the fact that these problems, as contrasted with the disorganized situations with which statistics can cope, show the essential feature of organization. And in fact, one can refer to this group of problems as organized complexity. So this is where it gets into maybe some of the things about cause and about, again, with the cyberneticists, they had this idea of causality, and they kind of ascribed some sort of purpose or some sort of intentionality to causality in a way that's very unsatisfying looking back at it. So he gets into this section here where he talks about what makes an evening primrose open when it does. Why does salt water fail to satisfy thirst? Why can one particular genetic strain of microorganism synthesize within its miniature body certain organic compounds that another strain of the same organism cannot manufacture? And he just goes through all of these things. And maybe these aren't very good questions. Like how do complex protein molecules know how to reduplicate their pattern? And is this an essential clue to the problem of reproduction of living creatures? So again, these are things that maybe we want to know how they happen, not just why they happen. And so again, we need to use statistical methods. They are not problems of disorganized complexity. They're problems which involve dealing simultaneously with a sizable number of factors which are interrelated into an organic whole. They are all, in the language here proposed, problems of organized complexity. These problems and a wide range of similar problems in the biological, medical, psychological, economic, and political sciences are just too complicated to yield the old 19th century techniques, which were so dramatically successful on 2, 3, or 4 year variable problems, or 2, 3, or 4 variable problems of simplicity. These new problems, moreover, cannot be handled with the statistical techniques so effective in describing average behavior in problems of disorganized complexity. So these new problems and the future of the world depends on many of them requires science to make a third grade advance. Okay, so this is where he's going over the next advance. And again, we're looking at this from 1948. So this was, kind of the point at which maybe people started thinking about complexity theory. And by from actually in the 1980s, there was a great run of chaos theory where it was like a very popular thing, it's fashionable to think about things in that way, and then it kind of receded. But this is where it's coming from, is this need to understand these sorts of problems. And so, it's interesting. I don't know if anyone's thought of writing a retrospective paper on this. But this is very much sounds like a Peter Holm type paper, where Peter Holm is the person who's written a lot of blog posts on sort of critiquing the history of complexity theory and what it accomplished and what it didn't. And this is kind of the idea where we can look back and see people calling for this thing that happened, and then the thing happened and it had failures and it made advances, and we can actually get a good sense of what that advance looked like as it unfolded. So that being said, I think complexity theory can solve some of these problems, but we've seen with complexity theory, especially with network theory and even with dynamical systems, that they can solve some of these problems that are open in this paper, but they can also not solve those problems. I think with networks, one of the big stumbling blocks was always causality. You can look at things that interact. You can build huge maps that show structure. You can get these average statistics of, clustering, or, you can do community detection, things like that. But you're still left with this fundamental problem of causality. What causes what? And maybe the lesson there is maybe things aren't as simple as they appear to be. Maybe things aren't as strictly, cause and effect as we like to think. So with networks, we look at a lot of interactions. We look at like A affecting B and B affecting A, and C, D, E, and F affecting B, and all of that. And maybe that's just the state of affairs. Maybe there isn't, maybe it's just a matter of representing that and then coming back to that problem with, say, like different statistical techniques that tell us maybe make estimates of how much like A is contributing to B, but also C through F. So, I think there's a lot here that I didn't attend the group, but he does get into a lot of this sort of stuff, and then he gets into the philosophy of science more generally, looking about what science is and what science isn't. I'm not really interested in that so much, but I do think, like if you're thinking about problems of statistics, problems of machine learning, problems of systems science, very generally and broadly, these have a lot of practical applications, and thinking about the different types of complexity that exist in the world and how we represent these in statistical models. It's very exciting stuff to me, anyways. So I don't know if we have any comments or questions on that. All right, so that's I think that's all for that. I think that was that was just kind of my take on that as well. I don't have any. Maybe next week I'll talk more about like the origins of chaos theory. I think that's really interesting as well. So another thing from our Slack. This was posted by Hussein, and I think Morgan commented on this. This is actually following up from the stuff that Morgan was talking about last week with the workshop. If you go back to last week's meeting, he talks about this workshop at Pitt, or in Pittsburgh, where they're doing things like bioprinting and cell culture and all this, and kind of putting together those pieces. And this is a paper that is kind of related to that. I mean, they're doing things with soft materials and development, although this is embryonic development. I don't know if, this isn't exactly organoids, but it's similar. And then doing this in the brain. So this is like very much this mind in vitro approach. So yeah, this is brain implantation of soft bioelectronics via embryonic development. So this is where they're putting together all these pieces to build things for the brain. So, developing bioelectronics capable of stably tracking brain-wide single cell millisecond resolved neural activity in the developing brain is critical for advancing neuroscience and understanding neurodevelopmental disorders. During development, the three-dimensional structure of the vertebrate brain arises from a two-dimensional neural plate. These larger morphological changes have previously posed a challenge for implantable bioelectronics to reliably track neural activity through brain development. So again, in development, you have these changes happening in the brain. So it's not just a matter of measuring like extant neurons fixed in space, but you have this sort of these large-scale changes in the morphology. So this is really kind of, if you implant bioelectronics, you have to account for these changes. So if you want to do something like track neural activity from, say, the neural plate structure to like the neural tube and then on to different brain structures, you're going to have to account for these kind of morphological changes. Here we introduce a tissue-level soft sub-micrometer thick mesh microelectrode array that integrates in the embryonic neural plate by leveraging the tissue's natural two-dimensional to three-dimensional reconfiguration. As organogenesis progresses, the mesh deforms, stretches, and distributes throughout the brain, seamlessly integrating with neural tissue. Immunostaining, gene expression analysis, and behavioral testing confirm no adverse effects on brain development or function, which is good if you want to measure brain function in situ, but you don't want to contaminate it by dysfunction. This embedded electrode array enables long-term stable mapping of how single neuron activity and population dynamics emerge and evolve during brain development. And then they use Axolotl as their model, so it not only records neuronal electrical activity during regeneration, but also modulates the process through electrical stimulation. So they're using an axolotl model of development, which is a common model in developmental biology, being able to take these implants, put them into embryonic axolotls, and then tracking brain activity through development. So this is a picture of sort of their model. This is panel A, where they start with stage 15, and they start with the neural plate, and they implant these ribbons into the embryo. Then stage 24, they remain in there as you get shape changes. Stage 26, you start to get the beginnings of the tadpole stage. Stage 40, this is the tadpole stage. You still have the implants in there. And then stage 46, you're getting into the more mature form of axolotl, and the implants remain in there. And you can see that, like, as the morphology changes, the implants are still in sort of the same place. You get these different types of three-dimensional changes, as well as shape changes, and it persists. Then we have, so this is in B, sort of this three-dimensional cartoon where we can see the three-dimensional aspects of the brain as it's differentiating. So you have the neural plate in B1. You have the implanted ribbons on the top of the neural plate. You have the mesoderm here and the notochord here. So you have mesoderm on either side, the notochord here, and the neural plate on top. And then, of course, the neural crest is where this neural plate invaginates. So that means that these implanted ribbons have to move down into this neural groove. So you have the notochord here and the mesoderm up here. And so everything folds downward. And so then the neural tube, as it's folded here, it's completely folded. The implants are embedded in here. You get the somites on these sides, the spinal ganglion. So the brain is building around the folded neural tube. The neural tube is closed. And then the neural tube closes and expands, and you can see the implants are still there with the surrounding tissues. And then 3 shows the three-dimensional distribution of bioelectronics. So as far as I know, they don't distribute themselves outward. They're just there. The ribbons are put down in the early neural plate stage, and then they get embedded by just the movement of the embryo. And so you get this sort of embedding, but they maintain a position relative to the things you want to measure. So if you want to measure things, if you want to have measurement accuracy, those implants need to correspond to brain tissues. And so they seem to be sort of, they seem to be consistent with what becomes the forebrain, and then the midbrain, and then a little bit of the hindbrain. So you can see this is the embryo brain here, where you have the 3-layered brain. And because you put the implants on the neural plate, they persist here, but they persist into these different regions of the brain. And then you actually have an I/O connector that remains outside of the developing brain. And this on the right here is the sort of the more mature, the tadpole brain, where you have this is the hindbrain here, the midbrain, and the forebrain. The electrons mostly are situated in the forebrain, and you have the I/O connector sticking out. Then you have some physics here. They measure the elastic modulus versus the molecular weight. So that gives you some performance information. Then you have stress, So you have cyclic loading, stress versus strain, and that's that. And they just show this throughout the length of the ribbon. J is force on neural plate versus different stages of development. So during implantation and development, where you get this, these electrodes have a certain force on the neural plate, so they have, they want to be able to make sure that it's not exerting too much force, it's not manipulating the way the neural plate develops, but it's also stable enough so that it doesn't shift as these embryonic movements are happening. And then K is stress on neural plate versus time as well, so you have this other function here. And that's basically, kind of the validation and kind of explaining how this works. And then this next figure here, figure 2, are the fabrication of tissue level soft mesh electronics. So, this again is a picture of the tadpole axolotl. So you have sort of showing the anchor and the IO pads and how this kind of fits into the the folded neural plate. This shows the neural crest and then the situation of these electrodes relative to the neural crest. Here are the implanted ribbons, and just showing this cross section. This is the fabrication of these ribbons. So you start with a silicon oxide wafer. You have the sacrificial layer. You have the spacers. You put these electrodes in. You have, so they're building up how they're fabricating this thing, showing the different components. This just shows a cross section of the different components. I think this is, it's electron microscopy of this in, I guess, it's implanted at this point. But basically it's showing the different layers as they exist in the electrode. And then M shows this is a photograph of the free floating mesh electrode array during stretching, bending, and twisting tests. So if you're moving with the neural tissue or you're moving with the embryonic tissue, you have to have a lot of flexible properties to the material. As you can see, it's a composite material. It's not naturally. It's made to be flexible, but it's not as flexible as the tissue. So it has to be able to bend and withstand a lot of stresses and strains and things like that. So you have the electrode, it can stretch, it can bend, it can twist, and they did this testing before they did the implant so that they knew that it wouldn't break apart as the embryo is making its movements. So, and then of course they have this electrical impedance resistance testing where you have measures of resistance and impedance and force for displacement, and then for over space and over days, over time. And then this just shows the neural plate and the neural tube, as we move through development, and kind of isolating that out and showing this process. You can see here that you have the implant at different stages of embryonic development. And there are things, you can do staining where you can see sort of the cells that it's contacting and other things like that. So you're using this red stain, you're using DAPI, and you're using this green channel to look at the different cells that it's contacting. Okay. So there's a lot of good information in this paper, a lot of detailed work, including the trace that you get out of these electrodes. So you're getting a lot of good information out of the brain. This is a proof of concept paper, so I'm not aware that they're testing any specific hypotheses, but they're showing sort of how the system basically works. I'm not sure how easy or hard it is to implement. In this figure, they do actually have some interesting results from kind of amputation studies. So this is where they have the implant, and then they amputate the tail in this group. But in this group, it's a sham. So they basically measure the firing rate of these neurons. They look at the embryos with tail amputation and without tail amputation. And so with tail amputation, you get this, after tail amputation in that group, you get a much higher firing rate than the embryos without tail amputation, which is interesting given what we know about regeneration, especially in embryonic development or in tadpole development. And then this shows, like in principal components analysis, what states these are in before and after amputation. So before amputation, you're kind of like, I guess, let's see, state 1, 2, 3, 4, 5, 6, 7. The darker circles are the states after amputation. The lighter circles are before amputation. So I guess before amputation are here, and after amputation are exploring more of this PC space. And that's consistent with this figure here. All right, so yeah, they talk about this a little bit in the results. We use the cyborg axolotl model to investigate neuronal activity changes following injury. So the reason they're doing this tail amputation is to sort of simulate injury. Specifically, tail amputation and subsequent regeneration. At stage 30, cyborg axolotl embryos underwent tail amputation, whereas the sham group remained uninjured. Continuous recordings showed that, following amputation, neuron firing rates increased significantly compared with pre-amputation levels, whereas sham embryos showed stable activity, as we saw in that figure. Tail amputation also alters neural population dynamics. Post-amputation neurons showed higher correlated activity, and factor analysis revealed shifts in neuronal states, with initial divergence followed by partial reversion. To test whether neural activity changes influence regeneration, we applied continuous external electrical stimulation through brain-embedded electrodes, mimicking post-amputation neural activity patterns. Stimulated embryos showed significantly accelerated regeneration rates compared with non-stimulation controls. By day 4, though, differences diminished as regeneration nearly completed. So this activity is sort of a consequence of a regeneration process. And we know that, regeneration often involves electrical signals in different cells or signaling. And so it's interesting that in the nervous system, of course, you're going to have this activity. Usually, it's like this, once you have amputation, it's usually the site of injury that's active, and there's some remodeling that goes on. There's some dedifferentiation of cells that happens so that you can have cells, instead of being differentiated, they become stem-like, and they migrate out and fill in the gap and do things like that. But it's interesting that there are also, I guess, cells in the central nervous system participating in that process. So that's an interesting finding. And again, they didn't do like these really specific hypotheses. They just did this sort of to show how the system can be useful. Okay, so why don't we move on here? This is an article from the Transmitter, and we've been talking about NeuroAI, I think probably for most of this year, last year. And some of the things that are happening in that community And, we also talked about sort of NeuroAI and embodiment. More generally, this is an article from The Transmitter by Bingwing Brunton and John Tuthill, entitled Breaking the Jar, Why NeuroAI Needs Embodiment. So the idea of the jar is basically the old philosophical idea of the brain in a vat. So there's this thought experiment that says, assume we had a brain in a vat. A brain was disembodied from your body, or from its context in the body, put in a jar, and if we just put the brain in a jar, could we have thought? What would happen? And so this is breaking the jar. So a lot of models of artificial intelligence, if even NeuroAI, they might involve some sort of biological inspiration, but they also exist in this jar. It's like the brain in the vat problem. And so the idea is that embodiment breaks us out of that jar and gives us something that is, body-like. So the idea is basically that if it's neuro-like, it has to involve some sort of body. And so then the question is what does embodiment look like? But that's maybe a different question, but maybe leading up to what they're talking about here. So brain function is inexorably linked or shaped by the body. Embracing this fact will benefit computational models of real brain function, as well as the design of artificial neural networks. Okay, so they kind of talk about the Teenage Mutant Ninja Turtles to start things off. Indeed, coordinating body movement is the ultimate point of brain function. So really, brain function is about coordinating body movement. You might say, well, I thought the brain generated language, or I thought the brain predicted things. That may be true, but there are linkages between, say, language and movement, language and the body. There are connections between language production and the body, obviously. And there is connection between sensory information and moving through the environment and using sensory information to shape movement. So movement is always at sort of the core of why we have a brain, or why our brains do what they do. The predictive aspect is sort of part and parcel of moving. So it's interesting that, when you reduce it to that, and sort of the coordination of that movement, moving through the environment, it becomes clear that we have to have an embodied model of brain function. The brain is not connected to the external world except through the body. It receives no sensory inputs except those transduced through sensory organs, and it can enact no consequences except through muscles and other actuators. Thus, the biomechanical feature and constraints of the body are built into the function of the brain. How animals move also determines how they gather new sensory information. Embodiment then is a concept that the function of the brain is inexorably shaped by the body. The embodiment lens is often neglected when neuroscientists study specific brain subsystems, particularly higher order cognitive functions. But in the growing field of NeuroAI, embodiment is now garnering greater attention. So Anthony Zador in 2023 proposed an embodied Turing test, which is this paper here, Catalyzing Next Generation Artificial Intelligence through NeuroAI. This is where they talk about the embodied Turing test, which challenges AI animal models to interact with the sensorimotor world at skill levels akin to their living counterparts. When they talk about a Turing test, it shifts the focus of these capabilities from those capabilities of game playing and language that are especially well developed or uniquely human to those capabilities, inherited from over 500 million years of evolution. Building models that can pass the embodied Turing test will provide a roadmap for the next generation of AI. So they still didn't mention what the actual Turing test was. But basically, the idea is that instead of focusing on performing well on games and performing well on these sort of combinatorial logic problems, that there is some connection to sort of motor performance and things like that. Okay, so some of the features of it is that animals engage their environments, they move around their environments, they explore their environments, and then animals behave flexibly. So if you you need to have as many inputs as possible to sort of enable flexibility. And then animals compute efficiently. So there's this energy aspect of the brain. So biological brains use relatively little energy to produce these kinds of behaviors that they do. And so these are all things that need to be incorporated into an embodied Turing test and allow these artificial agents to perform at levels similar to their counterparts in nature. Okay, so people have thought about this at least. And so here we discuss three key features of embodied intelligence that are characteristic of animal brains. The first is feedback, the second is biomechanics, and the third is modularity. We argue that embracing these three features will benefit computational models of real brain function, as well as the design of artificial neural networks that move beyond large-scale word salad generation to efficiently accomplish real-world tasks. Okay, so they talk about feedback being a ubiquitous characteristic of biological neural networks. Feed-forward models, in which information flows in only one direction, such as from the retina to the lateral geniculate nucleus to the primary visual cortex, are popular because they are easy to interpret. But that's not usually the way processing works. A lot of times it involves constant and multi-scale feedback, where you have fluctuating physical environments, recurrent connections, interlinked organs, and cells that modify themselves through gene regulatory networks. So these are all kinds of things we have to take into account. Again, we don't really make a statement about how much of the variation they contribute to, but they're in nature, and so we should replicate those. Second, biomechanical features of each specific body are essential to understand the function of the neural system within it. Consider, for instance, this video of a trout. The body undulates in the current and then surges upstream towards a small rock. This is what appears to be a dexterous natural behavior is, in fact, executed by a recently dead fish. So this behavior requires no neural activity and arises entirely from the interaction between the vortices shedding from the rock and the biomechanics of the fish body. So this is where you can actually replicate, or I should say, the movement of the body occurs with or without neural activity. So if you observe a trout moving upstream, it's maybe driven by external forces as much as sort of intentional internal forces. And so this is kind of an example of that. But this also speaks to the sort of mechanical or physical intelligence, which is where the body actually has, it sort of restricts the dynamics that are available to it, but also can be driven sort of by external forces. Another good example of this, of course, is the passive dynamic walker, which if you're familiar with robotics, you know what this is. It's a physical model of a walker, of a bipedal walker, where you have this robot this sort of inverted pendulum that has feet, and then you just push it along. You can push it externally, and the thing will walk like a bipedal organism, and it will eventually stop moving, but you can put enough input energy into it to get it to move and locomote, and it's very similar to, say, like a human walking. And so that's, again, another example of this where you don't need neural activity to drive this behavior, but nevertheless, you get this movement behavior out of a mechanical or physical system. So that's another point. And then third, our brains are highly modular. Even as we advocate for an integrative approach, there is undeniable value in deeply characterizing specific parts of the brain and musculoskeletal systems in isolation. But of course, these modules need to be stitched together in a way that's functional and gives us this sort of whole brain activity. But of course, modularity isn't just about dividing things into specific modules and then putting them together. Once you get to the systems level, modularity or modules can be a source of bottlenecks. So in the retina, retinal ganglion cells are the only cells that send outputs to the rest of the image-forming visual system, and motor control circuits all converge onto the final common output of motor neurons that synapse onto muscles. Such bottlenecks define modules, making it possible to develop computational models of each module that nevertheless interface with one another, and from which emerges the holistic behavior of the virtual animal. Importantly, models of different subsystems can be made at different resolutions and learn from different data. And so these taken together form this embodied perspective. But there is still a chasm between the aspiration of studying an embodied brain and how to implement it in practice. So, let's see. What makes the emerging zoo of virtual animals distinct from the previous generations of biomechanical models is in their compatibility with models of the brain. We can think of brain models as controllers that plan and execute behavior while responding to sensory stimuli from the environment. Of course, there's this model, Mujoko. When we've talked about Mujoko, which is a physics engine that simulates the biomechanics of skeletons, muscles, and tendons. This is based on kind of simulating robotic systems, but you can also use this for AI. A JAX accelerated implementation called Mujoko MJX now makes training whole body virtual animals tractable for researchers with access to GPUs. And so we need a good physical simulated environment. Mujoko is a candidate, but there may be other ways to do this. The prospect of developing a fully integrated neuromechanical model is perhaps closest to fruition for the adult fruit fly Drosophila. The recent completion of comprehensive synaptic wiring diagrams, known as connectomes of the fly brain and ventral nerve cord, mean it is now tractable to hook up computational models of neural dynamics at the resolution of cells and synapses to whole animal biomechanics. Such embodied models will open the door to in silico experiments that are currently not feasible in real animals, complementing existing genetics and behavioral tools. So consider the sensory control of robust walking. For more than a century of intense study, we know that nerve cord circuits generate rhythmic motor patterns autonomously, yet when the walking animal encounters a perturbation, such as uneven terrain, it must integrate feedback from sensory neurons with feed-forward commands to adjust, recover, and maintain walking. In silico experiments that ask how neural dynamics of recurrent circuits, coupled with biomechanical consequences in a virtual walking fly, complete with interactions with an unpredictable physical environment, will help us untangle these dual functions. And then, of course, they get into foundation models of animal brains and behaviors, and how those can be maybe useful, using the principles of embodiment to build these foundation models. So, again, this is a nice article because it kind of lays out this vision for embodied NeuroAI. I don't think it's anything that people haven't thought of before. It's just kind of putting it in one place. I think that's all for today. Did we have anything we wanted to talk about before we go? Or
Not really related to what this meet is, but I think like a few weeks ago I spoke about this whole Project X program that we have in our college. So yeah, that has started, and we will have a bi-weekly meet starting soon.
okay,
So once they start, I think I can put those up. I can bring those updates.
yeah, that would be great. Looking forward to it.
Yep. Thank you.
Yeah, no problem. All right, I think that's it for today. See you next week. Thank you.
Bye. Thank you.
Bye.